We are ready to start. Welcome everyone. Thanks for joining us today for the neuroelectrics webinar. We started with a little movie presenting our devices and our developments. So my name is Rafał Nowak and I will be again your host today. This is the series of neuroelectrics webinar, and this is actually the 12th from the series of neuroelectrics webinars. So actually, I just realized that this is our little anniversary. So we started our regular webinars already 1 year ago. So happy anniversary to everyone involved in the preparation of the webinar and everyone who follows our webinars from the very beginning. It's 1 year already. And today we'll be talking about using of multi-channel tDCS to improve bimanual motor skill learning. And I'm very happy to welcome our guest speaker, Dr. Nils Pixa. Hi, Nils.
Hello, everybody.
Good to have you here, Nils. So before we get started, some additional information. We'll begin with Nils' presentation. And this will be followed by a question and answer period. So please free to submit your questions at any time using the Q&A tool. And we'll record the presentation, so this will be available also offline. So please let me quickly introduce our speaker, Dr. Nils Pixa. Nils studied sports science at the Johannes Gutenberg University in Mainz in Germany, with a focus on the recreational and elite sports. After his diploma, he started his PhD at the Institute for Sports Science, still at the University of Mainz, in the working group of cognitive neuroscience in sport and exercise, led by Professor Michael Doppelmayr. And during his PhD, he focused on the tDCS effects on motor performance and learning, and primarily investigated the effects on bimanual motor skill learning by using multi-channel tDCS. After a postdoc position at the Chemnitz University of Technology in Germany, he's currently working as a postdoc at the Institute for Sport and Exercise Sciences in the Department of Neuromotor, Motor and Behavioral and Exercise, led by Professor Claudia Wilker at the University of Münster in Germany again. So again, obviously, a lot of interest shared with us at Neuroelectrics. Welcome, Nils. So the stage is yours.
Okay, perfect. So I will start with sharing my screen with you. Does it work?
Yes.
Okay, perfect. So yeah, thank you very much, Rafał, for the kind introduction. And also hello, everybody from my side here from Münster in Germany. Thank you very much also to Neuroelectrics for the invitation. It's a very pleasure for me to be here today and to present you some of my research about tDCS and bimanual motor skills. And yeah, let's start, I think. So yeah, why studying bimanual motor skills, or why is it so interesting or relevant to study bimanual motor skills and bimanual motor skill learning? First of all, it has some important functions in our everyday life and is an important activity function for everyday tasks that, yeah, at least we all know, I think, and perform a lot of these everyday activities. So, for example, opening a box to grasp something which is inside. That can be also nicely studied in an experimental condition, for example, as it was conducted here by a colleague of mine from Julian Rudisch. But we know also other everyday activities such as tying one's shoelaces, buttoning a shirt, and also more work-related activities. So, for example, a hairdresser giving you a nice new cut. And actually one prominent or very current example is to put on our COVID-19 mask. So I don't know whether you have ever tried to put the mask on with only one hand. It's quite difficult and looks maybe a bit funny. So normally we use our both hands just to put on our masks. Another point is that bimanual skills are important activities, functions for sports, especially in ball sports, for example. Here we see some volleyball players blocking a smash, But we can also imagine some basketballers passing a ball, or a rugby player who's running through the defense and trying to catch a ball. And yeah, there are also other sports like juggling, for example, which I think is a very prominent example of bimanual skills, and also swimming or rowing relies strongly on bimanual activity. Moreover, we know several neurological diseases or injuries to the central nervous system, which can lead to a reduction or decline in bimanual coordination. So it is thought there is also a relevance from neurotherapeutical purposes, for example, to regain these bimanual skills. And coming more from a research perspective, it is also a kind of model activity to study principles of motor coordination, for example, and also a kind of model activity to study neural networks and information processing in the brain and also between the hemispheres. So as illustrated here in these three small pictures, showing the bilateral activity or the increase in bilateral activity in the brain during the performance of bimanual tasks, and also the information flow or information exchange within and between the hemispheres. There's also some research about the frequency, meaning how often we use bimanual actions in everyday life, and that revealing that the abundance of daily activities are bimanually and occur more than twice as often compared to unimanual actions alone. And we mainly use our both hands to manipulate something or interact with our surrounding environment. From a more detailed research perspective, we can categorize bimanual actions quite similar to unimanual actions, and also other forms of movements. Shown here is a categorization scheme that is often used in, for example, movement sciences and also in the field of motor control. And this is adapted especially to bimanual actions. So that scheme can be found in a very nice review article from Celine Mas from Belgium. And the scheme divides bimanual actions into three categories. So discrete actions, which have a clear start point and ending, over to serial tasks where several discrete actions are performed in a certain series or sequence with a certain order, and also some continuous actions with no clear start and end point. Another non-manual example for that would be, for example, riding a bicycle. And moreover, discrete actions can be performed repetitively or in a non-repetitive fashion, and the complexity and the difficulty can be varied, for example, by increasing the range of motion, meaning, for example, the distance between the target points that need to be pointed or touched in that task here. And but we can also manipulate the size of the targets or the rhythm that is required to perform the task or the rhythmic activity. Similar modifications can be performed in continuous tasks, and moreover, others added or described bimanual action also as manipulatory or non-manipulatory, so depending on a bit on the task goal that we want to accomplish, and also more functional tasks, meaning more object-oriented or goal-directed. So functional can be also transferred or seen as more, let's say, related to everyday life activities, which is also shown here in the serial task, for example, where a person is grasping a small bottle with one hand and open it with the other hand. So that's a very prominent activity that we all perform throughout our days. Another very common classification scheme relies on the symmetry of arm movements. So whether we use our arms in a symmetric way or in a, let's say, asymmetric way, I can really show it here in my camera. and also depending on the task goal that needs to be accomplished. So bimanual actions can be performed in a symmetric, also in an asymmetric fashion, and both might include independent goals, meaning each side has an own goal, or a common goal, meaning an interdependency, which is required to accomplish a given task, for example. And that can be accomplished then in a parallel or in a cooperative way. And for example, a symmetric task with a common goal and which needs to be achieved in parallel could be something when we, for example, reach forward to grasp something. So our arms and hands are working in a parallel way. And a cooperative way comes then when we now pick up, for example, a box and try to pick it up and bring it to another point, for example. And they can be also seen for asymmetric actions. A comparable model to the one that is outlined before from Kantak and colleagues is provided by Wojtowicz and colleagues. And she classified bimanual actions also based on symmetry of the upper limbs, so comparable to the model of Kantak that was shown before. But she adds also a scale about the level of complexity. So the model shown before did not attach complexity. There was no differentiation between independent goals or common goals or symmetry or asymmetric bimanual actions, for example, but that's included here in that model. And moreover, the traditional classification of symmetric bimanual actions included. And it goes back to early work, for example, from Kelso in the 70s, where a lot of bimanual or a lot of finger tasks were investigated or under research to investigate that bimanual coordination aspect. And these bimanual actions can be further divided into in-phase, complex phasing. In-phase bimanual actions, meaning the involvement and activation of homologous muscles. For example, when I do something like this. And in contrast, antiphase movements include the activity of non-homologous muscles, meaning when I do, for example, something like that. Whereas complex phasing of bimanual action relies on both and on an irregular activation pattern, including homologous and also non-homologous muscles. And as shown here in that figure, in-phase and antiphase symmetric movements are seen as less complex compared to then complex phasing, and also in comparison to non-symmetric tasks, which can be either complementary, as also outlined by the previous model by Kantak, and also in an independent way that seems to be the most complex bimanual action, relying on that model here from Wojtowicz. So let's have a look on how our bimanual skills assessed. And that figure is also taken from the paper from Celine Maas and al. And it shows some kind of traditional or classical bimanual tasks, which are mainly used to assess bimanual motor skills and learning, and that reaching from several bimanual pointing tasks or some sequence tasks, for example. Also, circle drawing is often used to assess bimanual skills. Here is a figure of a well-known Purdue pegboard task that is often used in clinical settings, but also more sophisticated tasks, for example, here bimanual visuomotor tracking tasks, where controllers or where the participants are requested to track a certain line, for example, with that small cursor controlled by both hands, and that can be varied, or the complexity can be varied, for example, by variation or modification of that controller ratios or the frequencies of the controllers. Coming now to tDCS and bimanual tasks. When we have a look into the tDCS literature about motor control and learning, then we found mainly unimanual tasks were used, as indicated, for example, by that nice review and position paper from Buch and colleagues in 2017. And I think that is also one point that the main target in tDCS studies, so the brain area, which are mostly targeted by tDCS, are the left or the right primary motor cortex, as indicated here by that red circles, and the contralateral supraorbital cortices as reference placements, as indicated by the gray circles. And mainly conventional tDCS, meaning tDCS using two relatively large sponge electrodes, were used in the studies of motor learning and motor control. However, there are also some studies using different montages to target also other brain areas, for example, the cerebellum and also other sites. However, since we see that there is less knowledge about, or there was less knowledge about the effects of tDCS on bimanual motor skills, we conducted literature research about tDCS and bimanual skills. And that was done together with my dear colleague Bettina Pollock from Düsseldorf. And there we identified 13 studies, including 3 clinical studies with patients that address the effects of tDCS and bimanual motor skills. 2 of these studies I will present on the next slides. And we found further that mainly also conventional tDCS was used, targeting the primary motor cortices with stimulation parameters ranging from 1 milliampere to 2 milliampere and a duration of 10 to 24 minutes. Moreover, diverse bimanual tasks were used, reaching from bimanual sequence tasks, for example, also bimanual tracking, for example, and mainly single session studies were conducted, meaning the experiment took place on one day with one application of tDCS, either concurrent or in a close approximation to motor practice, and sometimes with a retention task, for example, after 24 hours. Others used also multi-session designs, meaning the task or the experiment took place over several days with several follow-up tests. However, no study used multi-sessions on one day, meaning, for example, application of 2 tDCS sessions within one experimental day. And all these studies, and I think that's a very good point, are sham controlled. I think it's very important from the methodological side. In the following, I will present you 3 studies that we have conducted in the context of tDCS effects on bimanual motor skill learning. And in the first study, we were mainly interested in the effects of on dexterity, especially on bimanual dexterity, in terms of fine motor control. Therefore, we used a well-known Purdue pegboard task, and the PPT can be performed into unimanual conditions and to bimanual conditions. The PPT consists overall of a board with 25 holes on the left and on the right side each. And the task is to place now as many pegs as possible into the board within 30 seconds. That in both unimanual conditions, starting with the dominant right hand and then the left hand, and also in the bimanual condition in a parallel way, meaning both hands are used to place as many pegs as possible into the holes. Additionally, another bimanual condition is the assembly task. I will show you in a second. And the assembly task meaning that these small montages need to build with the use of both hands, and each consisting of several parts. So 1 peg, 2 washers, and the collar in between. And the order is that the right hand grasps 1 peg, place it into the hole, while the left hand is grasping a washer, placing it over the peg, and then the right hand the collar, and then another washer, and then 1 assembly is built. And also the task goal is to build as many of these assemblies within the time period of 30 seconds. Since bimanual motor performance shows mainly bilateral activity, as revealed, for example, by several neuroimaging studies, we used multi-channel, or we created a multi-channel tDCS montage using 8 channels and using the star stem 8 with the pi electrodes to target both primary motor cortices. And yeah, as shown here on that figure, with that bihemispheric multi-channel approach, we wanted to target the left and the right primary motor cortex, especially the hand areas. So with our montage, we tried to spread the electric field coming from the electrode position C1, 2, and C2, and we wanted to spread it bilaterally to 3 return electrodes on each side. So we think that, or we mainly wanted to target the hand area. And as shown here in that simulation of the electric field, the peak of the electric field is mainly over the hand area. It can be also related to the electrode position C3 and C4, and spreads over to the reference electrode used in that montage. So the stimulation parameters were 1 milliamp per active electrode, so on C1 and C2, and the current was equally spread to the 3 return electrodes on both hemispheres. The idea of bilateral anodal tDCS is not new, I have to mention. It was several years ago introduced in a study by Gomez Osman and Phil Fold, namely in 2013, addressing also bimanual motor skill learning using a motor sequence task or a typing task. And they use also conventional tDCS, so placing 2 sponge electrodes on the left and the right primary motor cortex and 2 on the supraorbital frontal cortex, but also 2 anodes placed here and the reference electrodes placed on the supraorbital prefrontal cortex. Our study design included 6 experimental days and was conducted as a pre-post follow-up test design, meaning between the pre and the post-test, 3 training days took place, where the participants practiced the Purdue Packboard task, all 4 conditions of it, and were received concurrently the anodal tDCS or a sham stimulation. All experimental days were separated by at least 24 hours, and the follow-up test took place about 1 week after the post-test. In that study, 31 participants were included, all right-handed, and were randomly assigned to a stimulation group or a sham group. Yeah, coming to the results, here we see the performance development in the Purdue Packboard task conditions, here in the unimanual conditions shown on the left side, on the bimanual conditions shown on the right side. And the performance gained, or the performance is expressed in the mean number of pegs in the unimanual and the bimanual condition, in number of parts in the assembly condition, meaning the number of parts, so the pegs, the collars, the washers, which are placed or used to build the small assemblies, were counted. And our results reveal, first of all, that all participants improved their performance in all these tasks, from pre to post and also to a follow-up test. And we see for the unimanual right-handed condition a significant interaction, meaning that the performance gains in the stimulation group were higher. There were stronger improvements in the stimulation group compared to the sham condition. And we see a further increase from the post to the follow-up test in the stimulation group, meaning that the consolidation of that task, that the motor learning, was increased. A similar pattern was observed for the both hand or the bimanual condition for the parallel fashion. And here we see also an interaction effect showing that the stimulation positively affects also the learning of that bimanual dexterity task. However, for the left hand and also for the maybe more complex assembly task, there was no significant effect from the stimulation. Yeah, coming to the second study, in that we were interested in whether we can transfer our findings from a Dextery's fine motor control task that was, for example, that was used in the Purdue Packboard task to a more cross-motor but still dexterous task, and investigate the effects of our tDCS multi-channel approach on sport stacking. So yeah, maybe some of you already know sport stacking. And I will show you the task with a quite fascinating short video from the 8-year-old Austin neighbor. He's now a bit older, so the video is a few years old. And he performs sport stacking, I think, on a very impressive and on a very high level. So have a look on this.
Hello, my name is Austin Neighbor. I'm 8 years old. I'm in the 3rd grade. I'm from Bloomington, Indiana. This is the 333. 1.816 seconds. This is the 363. 2.275 seconds, which is a new 363 world record for an 8-year-old. And this is the cycle. I'll redo that. .299 seconds. That is a new 8-year-old world record.
Yeah, so cool, so amazing. I think it's so cool when he said, "Oh, I will do that." It's so quite nice and very amazing. Nice task, and it makes a lot of fun. And yeah, all of our participants were quite motivated to perform that task, and we also have some small competitions in our working group. But yeah, small spoiler, no one of us and also no of the participants reached that performance level, for example, as here from shown by Austin Neighbor. And yeah, coming to that study, here we included 32 right-handed participants and also assigned them equally or randomized and equally to 2 groups, simulation group and a sham group. And we used the equal multi-channel tDCS parameters and montage, as well as the same study design as before in the study where we used the Purdue Backboard task. And now the participants practiced 2 different sport stacking formations. On one side, here shown in the figure A, the 3-3-6-3, and in B, the 1-10-1, meaning that the formation has to be built up or stacked up and stacked down as fast as possible. Yeah, that was then counted in the pre-test and the post-test and also in the follow-up test. And on 3 training days, the participants practiced both stacking formations and received concurrently tDCS or sham tDCS. So same as in the Purdue Backboard task study. Yeah, coming directly to the results, that revealed significant improvements in stacking time, so meaning how fast the stacking was performed over the time course of the experiment, expressed here in that figure, A and C. And further, we found stronger improvements in the stimulation group for the 3-6-3. However, that was not the case for the 1-10-1 formation. And further, we separately analyzed the performance gain score, meaning the difference from the pre-test to the post-test and to the follow-up test. And still here we see a significant difference between both stimulation conditions with a higher performance gains or a stronger improvement due to the bilateral or bihemispheric application of a nodal tDCS. Although there is a kind of mixed finding, since we found no significant effects on the other, namely 1-10-1 stacking formation, we could show that our approach seemed to have a positive effect also on cross bimanual actions, and that we partially can transfer our findings from the fine motor control and dexterous task also to a more cross motor bimanual skill. Yeah, coming to the last study that I will present you today here in this webinar, we investigated there the effects on tDCS on bimanual tracking task that relies mainly on fine visuomotor control and precise spatiotemporal interplay between both hands. And the task is shown here on that figure. It's a modified figure of the so-called two-hand task, where participants are requested to guide a cursor, here shown as a red dot, as fast and accurately as possible through the course here presented by using either 2 joysticks or 2 rotary buttons, and the right joystick and the right rotary button controls the cursor in a vertical way, and the left-sided controllers control the cursor in a horizontal way. So there's, yeah, as I said before, there is a nice and precise spatial-temporal coordination between both hands required to accomplish that task goal as accurate and as fast as possible. And here again, we use our bilateral anodal tDCS approach. And now further, we extended our multi-channel application to the parietal cortex, which is also known to have a prominent role in bimanual coordination, for example, due to the multi-sensory integration aspects, and also to bimanual motor skill learning. And here we used, again, as shown here in that STIM view, again, 2 active electrodes, 2 anodes placed over the parietal cortex, so namely the central parietal side and the parietal side. And now here we used 4 return electrodes in total to spread the electric field bilaterally over the parietal cortex and the targeting the precuneus and also some parts of the posterior parietal cortex. So I have to add that we used the same stimulation parameters. We used also 1 milliamp per active electrode and the duration of 15 minutes for the application. Moreover, we slightly modified our study design compared to the both studies that I have presented before. And we have now 5 experimental days, including 3 so-called test and training days, so T1, T2, and T3. Those were separated by at least 24 hours, a maximum of 48 hours. And now the pre and the post test took place on the same days as the motor practice, and the motor practice was also with a concurrent tDCS application. So here we see these test and training days, and we have some 2 additional retention tasks, one 24 hours after the last training session and one approximately 1 week after the first retention task. So important to note is that the task was practiced only with a joystick condition with the application of 50 minutes of these multi-channel tDCS, but the task was also assessed with a rotary button condition in the pre-test, the post-test, and both retention tests, meaning that this rotary task condition was used as a transfer task and therefore was only assessed in these testing sessions and not practiced. Coming also here to the results, first to the mean error rate, so meaning how often the participants went out of the course. And in short, we could not find any effect of any tDCS condition on the error rate in the 2-hand task. And also, all participants decreased the error rate over time, showing also in the rotary buttons condition that was also assessed. It was only assessed in the testing sessions and was not practiced. And that's a very impressive finding, I think, that also the transfer condition was seems to be learned over time, or the performance in that was increased over time. However, it was not affected by our stimulation approaches. In the second parameter that we analyzed, we looked on the mean movement time, or on, again, the speed of how fast the participants were able to guide that cursor through the course. Again, for the joysticks condition on the left side and the rotary buttons or the transfer task on the right side. And here we see again significant increase in the performance, so a significant decrease in movement time, faster movement time, faster speed in both conditions. And moreover, we found a time by group and task modality interaction, so task modality meaning joysticks and rotary buttons. And further analysis revealed also two-way interaction between group and modalities. Therefore, we did some follow-up analysis and found some differences between the stimulation of the primary motor cortex and the sham condition compared to parietal stimulation that was observable here in the second retention task and in the rotary buttons condition, also the transfer task, on the second retention test. And this difference was interpreted as a significant decrease in the performance performed or using the rotary buttons, and might be attributed to a deterioration in the task transfer due to parietal stimulation, and maybe a reduced or a negative effect, can also interpret as a negative effect on consolidation of the transfer task, since it was only observed after the retention interval of 1 week. Moreover, we were also interested in the online learning effects, meaning the improvement from within one training session, from the pre-test to the post-test on each day. And here we found significant time effects, so meaning the improvement, there was an improvement due to the training sessions. However, it was not affected by that stimulation. And in another analysis, we looked on the offline learning effects, meaning the difference from the training, from the training days to the consecutive day, and or to the also to the retention task. And here we found also significant effects for time, so significant improvements. And yeah, not surprisingly, the interaction occurred also here, showing significant differences between the stimulation conditions of the primary motor cortex, or the stimulation of the primary motor cortex, and sham condition in comparison to the stimulation of the parietal cortex. Yeah, taken together, the results revealed that multi-channel tDCS targeting bilateral primary motor cortices with a nodal tDCS seem to increase unimanual and bimanual dexterity, so more fine motor related tasks. It can also partially enhance learning of a more cross-motor bimanual task, as revealed with the sports stacking study, but it has no effect on bimanual tracking that can be associated strongly with bimanual coordination, also relies partially or maybe more on visual motor control. However, parietal multi-channel tDCS seems to have no effect on bimanual tracking, since we see no improvements due to parietal tDCS, but it seems to have a slightly negative effect, or might impair bimanual learning transfer in the long run, affecting consolidation. One might speculate about task dependency of the effects, since we see some mixed results between, but also within the studies. For example, in the Purdy-Packboard study, we found effects on 2 subtasks of the Purdy-Packboard task, but not on all 4 conditions. And also there were partially effects in the sport stacking formations. And these task dependency of effects is also seen, as I would interpret it, also for unimanual tasks and other motor learning paradigms used in tDCS literature. And I think that there is the need for a more systematic exploration of these effects in the future, and also addressing different kind of tasks and comparing different kinds of simulations. I have to mention also some limitations of our studies. On one side, we did not include a cathodal condition, so we compare our results not to a cathodal tDCS condition that might also provoke some effects. And on the other hand, we did not assess any physiological data, so we cannot rule out our stimulation effects or the effects for our multi-channel tDCS application on the neurophysiological level. Yes, and I'm at the end of my presentation, and I think there is nothing more left to say than, first of all, thank you very much for your attention. And my special thanks goes to my colleagues from the Institute of Sports Science in Mainz, especially to my supervisor, Professor Doppelmayr, and all the other great colleagues and persons where and with I have done all that work that I have presented today. And yeah, if you have further questions or comments or additional things, please drop me an email, would be highly appreciate. And here are my references. And again, thank you very much for listening. And now I'm looking forward for your questions.
Thank you very much, Nils. We are really impressed. I especially have to see the video of the stacking task, I think, with this kit in slow motion version. You provided the link to the YouTube video, so we'll check this again.
Yeah.
So we are ready for the questions from audience. You can use this Q&A tool to ask questions. But let me ask some. So the natural question would be, what about applications? So I would expect to translate this already, and I would see, for example, kids with cerebral palsy, with the improvement in motor learning abilities, for example, in cerebral palsy directly. So do you think this will be like easy transferable to the clinical application?
Yes. I would agree, or I would say yes, it is transferable also to clinical applications, since there are several studies, for example, revealing that also bimanual therapies or bimanual tasks used in the rehabilitation of stroke patients, where we have only a hemiparese, for example, can increase the rehabilitation process, and it might be also beneficial to combine that with tDCS, for example. And I know a recent study in cerebral palsy patients that used sport stacking for rehabilitation purposes, for example. So I think, yeah, there is a field of application to combine bimanual training and tDCS also in clinical or for clinical purposes.
Okay, thank you very much. So we are going to some questions we have already. First one from Bilal. Any research using both hemispheres stimulation? So actually, if I understand correctly, you were stimulating both hemispheres in your studies. So actually, this is
yes. We stimulated both, and as I said, that as far as I know, there is one other study that was conducted by Gomez, Osman, and Field, and they also used a similar approach, but used conventional tDCS by attaching two large sponge electrodes on both hemispheres and concurrently using two reference electrodes on the supraorbital frontal cortices as references. And they use also a nodal tDCS and compared their results to sham stimulation condition and used a bimanual typing task.
Okay, so actually you're answering the second question from Vera. Thank you very much for interesting talk. Do you have a direct comparison in, I understand, between multi-channel and classical two electrodes montages? So.
I interpret that question in the direction that we compared that, for example, the stimulation approach used by Gomez Osman as outlined before, or other approach, and no, we did not. We very soon decided to use that multi-channel approach and that high definition approach, or using the pi electrodes to have a more focal stimulation. But that would be a very interesting thing, I think, to compare conventional tDCS that is also done by unimanual stimulation, so to say, where we know some studies comparing, for example, the 4x1 high definition montage to a conventional tDCS montage. And it would be also quite interesting to compare conventional bilateral or bihemispheric tDCS with our approach. Yeah.
Okay. So let me only here do a reference to the study. Actually, we're participating with a network targeted stimulation. Actually, this was just to show the difference between bipolar stimulation with the comparison of bipolar stimulation and network stimulation. Network of motor cortex has been defined using resting state fMRI, and then based on the definition of seats of the network has been used as the target for the optimization. And this is Fisher et al. I will send you the reference, and you can find exactly the montages, the optimized montage, the current targeting in the optimized way the motor cortex networks, because the question would be, what was your motivation to use this specific montage with two electrodes over motor cortex, and then in the temporal part, the free reference electrodes?
Yeah. So I know the study from Fisher, and it's very nice that you mentioned it. It's a very nice study. Really appreciate. And our rationale was, so we screened the literature, so mainly the neuroimaging literature, using bimanual tasks and different forms of bimanual tasks. And there we identified that both primary motor cortex are increased during bimanual performance and also have a prominent role for bimanual motor skill learning. And therefore we decided to target both primary motor cortices with a nodal tDCS. And then we used the STIM view software to model our electric field. Considering several parameters, for example, the distance between the electrodes and also the placement for the electrodes, and come then to that conclusion that our approach fits nicely to the orientation of the electric field that we wanted to produce to target then the hand areas, which are mainly based underneath the electrode position 3 and C4. But there are also some simulation studies showing that the main impact of the electric field is not right underneath the electrode, but between or somewhere aside to the active electrode, to the anode, for example, in reference to the return electrodes. And that was our approach to target the hand areas on both sides. Yeah, but it would be more beneficial to have own, let's say, neurophysiological data, as it was conducted in the Fisher study, using a task, using fMRI, for example, or another neuroimaging method, and then to identify that network, and then just modeling based on that network data your own montage or application. Yeah.
So now actually you have very good results. So the suggestion only actually to tune up and to remove variability that you can get with different kind of stimulation.
Sure,
Thank you. The question from DN: is there any study about improvements of learning in bimanual skills in neuroprosthetics users due to tDCS? Would this be useful in such a study, do you think?
To be honest, I did not come across such a study, and I'm not too much into the topic of neuroprosthetics, so I cannot really say about it. I didn't see any research about it, but it would be also an interesting research question, I think. Yeah.
Okay. A question from Bailal. Any research comparing sport performance related research using 2 milliamperes versus 4 milliamperes? So you are not getting to 4 milliamperes yet.
Sorry, I think I didn't get the question right.
The comparison between 2 milliamperes and 4 milliamperes per electrode with the current. If you have any experience or you know about any studies that were comparing 2 milliamperes versus 4 milliamperes current.
I think the current intensity and also the current density then, in that it mainly depends on the montage you are using and also the electrodes you are using. So as far as I know, the small high definition tDCS electrodes, so here the pi electrodes, can be used up to 2 milliamp without then inserting stronger sensations. So then it might get a bit more uncomfortable. But there are studies that increased the current intensity to up to 4 milliamp by using sponge electrodes, and that seems to be safe and can be also used. But we have also to consider whether there is a linear relationship between increasing the current intensity and the effects of tDCS. And I would say there are several studies that did not show such a linear relationship. There might be another kind of relationship. So I hope that answered a bit that question.
So the simple actually argumentation would be, well, why we do not increase the current and we not increase the duration of the stimulation, but that you are mentioning this is there is a nonlinear effect after crossing some thresholds, and it's not so simple. So that's why it's even better to use more focal stimulation with less intensity than increase the intensity.
Yeah, that is one point that I would totally agree to. To look on the focality and also on some kind of network stimulation, for example. So define your target or find your target. You want to increase the excitability, also decrease the excitability, and then I think in the next step, think about the duration of the stimulation and also the intensity of the stimulation. Yeah, but there are a lot of question marks still, and still outstanding research questions, I think.
Next question from Anke, actually, that you are answering right now. Thanks for the nice talk, Nils. I was wondering if you can tell a bit more about your choice of montage. You focus a lot of M1, but the promoter area, PMC and SMA, are also highly relevant for bimanual control. Any plans to place the return electrode in a way that will spread the field to also include premotor cortex and somatosensor motor area? Supplementary motor area. Supplementary,
yeah. Thanks for that question. That was something we discussed a lot about, and then decided first to target the primary motor cortices. But I think the next step could be to target the premotor cortex, which is strongly involved in motor learning. But maybe can be not really so to say, it's not so easy to have such a kind of focal stimulation just to target the primary motor cortex or the premotor cortex, because they are so closely together. But I think there would be an approach that might be very beneficial to use a more focal stimulation, so high definition stimulation, to precisely target the premotor cortices and also the SMA. But in our studies, we decided to use to stimulate the primary motor cortex, and then extended our stimulation to the parietal cortex, which is also known to be prominently or substantially involved in bimanual coordination, and it can be very well divided or separated to the effect on the primary motor cortex. However, I think we need to consider that it's an outstanding question whether we can easily transfer the excitability change that we see in the primary motor cortex due to tDCS also on other non-motor regions. So for example, the parietal cortex. I think there's also something we need to consider.
Okay, perfect. Next question from Hugo. Very nice presentation. Just curious about motor performance differences at baseline in the studies that you presented. If there is a difference, did you account for baseline differences over time?
Yeah, I didn't mention it, but yes, you are right. Especially in the sports stacking study, we have some difference in the baseline performance, and we account for it. We control for it by using an ANCOVA. After controlling for the baseline difference, there's still the significant interaction effects for the nodal condition. Yeah.
Okay. Next question from Bettina. Could the participants feel the stimulation? Was it able to double blind the studies?
Yeah. Also something that I didn't mention, sorry for that. Yes, was double blinded. The experimenter and also the participants was not able to know which kind of stimulation is applied, so whether a nodal or sham stimulation. And we let the participants rate what kind of stimulation they would have received. And there was, by chance, so they did not have a clear idea whether they received one or the other stimulation. And another point is that in the last study, where we used also the parietal stimulation, the sham condition was equally divided to the between the primary motor cortex stimulation and the parietal cortex stimulation. So half of the participants received sham stimulation over the parietal cortex, and the other half the sham stimulation over the primary motor cortex, just to have an equal distribution between both stimulation sites.
Okay, thank you. A question from DN. Is it possible to simultaneously record the EEG data while doing tDCS stim, or will the EEG be very disturbed due to the stimulation and so would be of no use?
Yeah, we did not use this in these studies, but I think that would be also a nice thing. One aspect I have to mention here in our study is that we used 8 channels for the stimulation, so there were no channels left that we could use for simultaneous EEG recordings. There is only the possibility to measure EEG activity prior and after the stimulation. That would be one case. And due to the aspect of the disturbances, yes, there are disturbances due to tDCS and artifacts, but I think nowadays there are a few nice algorithms and ways to discriminate the real oscillatory activity from the activity that is induced due to the tDCS. It would be much more complicated when we use tACS, for example, and then to separate the original oscillatory activity from the brain from that is induced on the head surface due to the tACS application.
Okay, thank you.
And there's a lot of research ongoing in that field.
Comment from Vera. Fisher et al. Use 4 milliamperes total inject current. Yeah, actually, so let me answer this. It's total, when in our devices, 4 milliamperes means that the current is splitted over all electrodes. So it's still 2 milliampere per electrode. But if you are using 8 channels, the maximum current that you can use is 4 milliamperes. Yeah. So this is explained in the article. Next question from the end. Good reference for studies about the safety of tDCS and limitations. So I can here reference also the guidelines, for example, where Andrea Antal is the first author, low intensity transcranial direct current stimulation guidelines about the stimulations. You can find this in clinical neurophysiology.
Yeah.
Next question from Bashar. Thanks for this interesting meeting and presentation. Does combined tDCS with skill training simultaneously have same effect of using tDCS alone on skills performance? What is your advice? So this is the all standing questions: before, during, or after.
Yeah. That are some very interesting and outstanding questions, I think, that need to be answered. And when we look into the literature, we see, I would say, both. We see that tDCS applied prior to motor practice has positive effects. We see that tDCS applied simultaneously to motor practice has positive effects. As far as I see it, that tDCS applied after motor practice seems to have not such beneficial effects in comparison to both other application timings. And when you look in the literature, for example, to the publications from Janine Reis and also from Britta Fritsch, they used also animal models to investigate that question. And they found, for example, that it is more beneficial to apply tDCS during motor practice or during motor performance, since there might be less activity that can be induced into something that is not going on or not working. So the neurons that are just in a kind of resting condition or resting state seems to not to be as much affected as neurons that are on the work, for example, when we do our tasks. And also that what was addressed by Rafal and the Fisher study, they used the motor task to identify the network, and that would not have been identified when we just let the people sitting there and rest and doing our measures. So my choice is, or my advice is, to use tDCS during motor practice. But I know a lot of very, very good research and very good papers using tDCS, for example, prior to motor practice, and has also some beneficial effects. And
actually, you have with your colleagues a paper in scientific reports in Nature that you find results only with aerobic exercise with the inhibitory effect of cathodic stimulation, and no with another, and no without aerobic exercise actually. So this is the good example of enhancement during the specific task, but for this specific task and for this specific region, and for inhibition, I understand, because so that will depend also on the task, on the and what you want to modify.
Yeah, definitely. And I think we have also to distinguish between motor task and also cognitive tasks. So in that recent study that we recently published in Scientific Reports, as you mentioned, we target the cognitive domain. We target executive functions and especially inhibition abilities by prefrontal tDCS and aerobic exercise and then a combination. And there we found that effects only when it is combined. So the participants were sitting on a bicycle ergometer, doing the aerobic exercise on a moderate intensity, receiving concurrently tDCS to the left prefrontal cortex, and doing the task on the same time. And there we found that effects. We found that effects, or we could not observe those effects in a very comparable study that we published a couple of months ago, where the participants performed the cognitive task prior and after the tDCS and the aerobic exercise application. And I think that's also a good example on the timing of tDCS to a task, for example.
Okay, so almost last one, clarification for Juan. What is the difference between reference electrode and cathodal TCS during bilateral nodal stimulation? Are cathodal and reference the same?
In my case, yes, since the active electrodes we used only anodal stimulation on the active electrodes on the primary motor cortex. So all other reference electrodes, or you can also say return electrodes, were cathodes. So when we flip the stimulation conditions to a cathodal stimulation targeting primary motor cortices, for example, then all the other reference electrodes would then be anodes. Yeah, but it depends on your montage that you are using. And for example, in the Fisher paper that I can highly recommend as well, you see there are also different forms where you can place your anodes, also cathodes, and depending on the network or your stimulation targets.
Yeah, but so if we are talking about anodes, the other electrodes will be cathodes always. And if you are talking about cathodes as active, so the other electrodes will be anodes, because the sum of the current we will have to have zero always.
Yeah.
Remember. But yeah, and the last questions, actually, you cannot escape from these questions from Matthias. How much could be the intensity in milliamps in time duration to try to study and enhance speed in canoa sprint? So those are the questions that we cannot answer yet, but I would like to know your opinion regarding availability of the consumer use devices. So we know that they are available already to the public. There are numerous professional athletes even that claim that have adopted tDCS during their training. So what is your opinion regarding this use?
Well, I think I see that a bit more critically. I see a lot of research going on to increase professional athletes by the use of tDCS, and on the other side, I see also an increasing do-it-yourself community. So I can also recommend the papers from Anna Wexler, for example, from the US. She investigates a lot about in that research field. And since I think tDCS should be also always used by professionals, by experts, knowing the parameters, knowing the guidelines, knowing also the contradictions and contradictory factors that need to be taken into account to prevent serious adverse effects that might emerge to a misuse of tDCS, I would say. And we should also have a view on the ethical aspects in terms of neurodoping, for example, when we use tDCS for neuroenhancement in high performance athletes. So my view is a bit critical. I think it's interesting to see whether we can bring high performance athletes to a higher level, but the ethical aspect is still something that we need to consider. And to the question, I definitely cannot recommend a certain intensity level and also a certain duration for that. I think one starting point when you do some studies in that field is to look into the literature, look into what was used in other tasks, maybe in tasks which are quite comparable to the task that you want to use or the performance that you want to increase, and work with that, do a nice literature research, and otherwise contact experts, go into discussion with experts, but then more in a concrete way, I would say, and always have that ethical issues in mind. That's my recommendation.
Okay, thank you very much. Actually, linking with this, so you are giving us an idea to organize next webinar and somehow to have a topic regarding ethics, regarding neuroenhancement, regarding neurodoping, do it yourself, over-the-counter use, and we'll try to invite some people from different fields, philosophy, ethics, and neuroscience to discuss also this topic during next webinar.
Yeah.
Okay. So thank you very much, Nils. Thanks for excellent presentation. This was the last question. I hope you will join us again for next webinars. We recommend to visit our webpage, neuroscience.com, where you can find more information about the upcoming webinars. Hopefully you will have a great day, and we'll see you next time. Do you want to add something, Nils, at the end?
Yeah, thank you very much as well for listening, for your attention, also for the very interesting discussion. I think if we have time, we could discuss, definitely more longer, but thank you very much. And also thank you very much again for the invitation and for the opportunity to present you some of my research. Thanks.
Thank you very much. Thanks. Goodbye.
Have a good day.
Have a good day. Bye.